Hello everyone, welcome back. This is going to be our last lecture video regarding enzymes. And in this particular video, we're going to discuss how we can control the rate of an enzyme reaction. And when I say control it, our focus is mainly going to be to slow it down as well as to perhaps stop it. Alright, so there's going to be several factors over here that can affect the rate at which an enzymatic reaction proceeds. So that's a reaction that is being controlled by an enzyme. Of the four, the first three should be kind of common sense. The first one is temperature and pH. So as we've already learned, that if we change the temperature or pH to an extreme that the enzyme does not like, it is going to denature, break down, and the rate of reaction is going to slow down, if not stop completely. The second would be the concentration of the enzyme itself. So if you have very little enzyme present but a lot of substrate, you're not going to make a lot of product, so the rate of reaction will be fairly slow. If you increase the amount of enzyme, then the reaction rate will begin to increase over time also. The third would be the substrate concentration itself. So if you have a lot of enzymes but just a little bit of substrate, well, you're not going to make too much products, correct? So the rate of reaction will be slow. To increase it, you would then increase the amount of substrate. One thing I would ask that you just realize, though, is that a rate of reaction, so let's just look at my little laser pointer. A rate of reaction, no matter how much enzyme or substrate you add, isn't going to keep doing this and going up in a straight line exponentially. Eventually what's going to happen, your rate of reaction would increase and increase, but eventually you would reach a plateau where you would just stay horizontal for a while. That's because you can't exceed 100% efficiency. No matter how much more enzymes or substrates you add, you're just going to reach a maximum rate that you can't make any more product. It's just not going to be feasible. And the last thing that we're going to focus on mainly for the rest of this lecture is going to be, are there any of what we call inhibitors present? And an inhibitor is going to be some type of molecule that's going to interact with the enzyme. So it's not going to be the substrate, it's going to be something else. And it's going to interact with the enzyme and slow it down or prevent it from actually doing the reaction that you're wanting to try to get done. So let's look at the first of three particular types of inhibition. The first one is going to be called competitive inhibition. Before I even pull up anything else on the screen, let's look over here at the little cartoon. All right, so the enzyme is in yellow, the substrate is in blue, so what's this thing? That's going to be the inhibitor. All right, and it's going to be what we call a competitive inhibitor. Because what competitive inhibitors do is they actually compete with and fight for the active site along with the substrate. Because they can bind to the active site because they're either exactly the same shape or close enough that they can get in there and clog it up, like putting chewing gum in a lock. So this substrate now cannot bind to this enzyme, so you're not going to be able to make enough product as a result. All right. Generally, you would not clog up every single enzyme. Some enzymes would still be working. However, clog up enough of them to prevent substrates from binding, and you can cause a problem within a cell or a body. So competitive inhibition, again, is going to be when a molecule, besides the actual substrate, can bind to the active site. All right, this will then lower the amount of enzymes that are capable of undergoing a reaction, so your reaction rate is going to start to decrease. And competitive inhibition use inhibitors that a lot of times we know them as poisons. We've heard of poisons before, right? Or toxins. You get bit by a poisonous snake or a poisonous other spider or some other type of animal, that venom can cause you to die. Well, a lot of times that venom have enzymes in them, or I'm sorry, have inhibitors in them that will interact with some enzymes in your body that are responsible for doing a very important function, like breathing or sending neurological signals from your brain to the rest of your body, and that leads to death. So a lot of poisons, such as the most famous one that's been written in movies and books all the time, is cyanide. Cyanide, if you've ever seen like a spy movie where they bite down on a tooth and they start foaming at the mouth and they die, well, what cyanide does is, when you expose it to water, it becomes a gas. That gas can travel to your lungs, where it'll block the enzymes in your lungs that absorb oxygen from the air. So it causes you to suffocate to death fairly quickly as a result. Cyanide isn't really used anymore in the gas chamber, one of the forms of death penalties that a lot of states had used in the past. Most times we don't use that anymore. We use lethal injection, which is they put you to sleep first and they inject you with a chemical that does something similar but stops your heart from pumping. Any of them, though, are competitive inhibitors. If you look over here, again, they bind to the active site and keep the substrate from binding. If the substrate is bound first, though, they can't bind. That's why you're never going to block every single enzyme. Another way to look at it here, again, here's your active site. Normally the substrate would bind to produce products, but now the inhibitor is going to bind to the active site like chewing gum in a lock and block the substrate from binding. The next type is called allosteric inhibition. It used to be taught as non-competitive. That's how I learned it as a student, or when I was in high school as well. But don't even bother writing that down. Let's just call it allosteric, because I don't want you to get confused and think that it is competing. All right, now what an allosteric inhibitor does is it is also, again, going to slow down or stop the rate at which an enzyme is working, but it does it differently. This inhibitor, whatever it is not going to fight for the active site. It's looking for another spot on the enzyme where it can bind to. That's called either the regulatory or the allosteric site. So if I bring your attention over here, all right, here's an enzyme. Here's a competitive inhibitor that's blocking the active site from the substrate. But you see this other spot? That's the allosteric or regulatory site. Now an inhibitor that's allosteric can bind there, and when it does, it changes the shape of the enzyme, including the active site. So you see how these little folds have now come in? That's going to keep the substrate from binding, where normally they'd be more facing outwards where the substrate could. So you're still inhibiting, but you just don't have to fight for the active site if you're an allosteric inhibitor. So ultimately what it's doing is it's changing the shape of the enzyme, the active site, so the substrate can't bind. Again, you're looking down here at this animation. All right, if the inhibitor binds to the allosteric site, the active site changes, the substrate can't bind. But if the substrate binds first, it would change the shape of the allosteric site, so the inhibitor can't bind. So again, even with allosteric inhibitors, you're never going to block every single enzyme. All right, you're just going to block enough of them to slow down the rate of reaction, or hopefully not stop it. Another graphic here. So here we go for allosteric inhibitors. It's going to bind to this other site here. All right, that's not the active site. When it does, changes the shape of the whole enzyme, including the active site. See how it's not a triangle anymore? It's this shape. Substrate can no longer bind. And the last type of inhibition, we're going to call this feedback inhibition. So if you hear that word feedback, that means something's coming back, right? Well, feedback inhibition, honestly, is allosteric inhibition. So whatever the inhibitor is, it's going to behave the same way. It's going to look for this regulatory allosteric site. But this version of inhibition, feedback inhibition, is actually going to use the product of a reaction as the inhibitor itself. So whatever the product is that's being made, if you make too much of it, it can actually come back and act as its own inhibitor. So if I bring your attention over here, let's say we're taking some yellow gumdrop candies, they're the substrate, and from this enzyme, they're being turned into candy corn. All right, I don't like candy corn. All right, so one or two are good, and then you're like, ugh, afterwards. Well, what happens if you make too much candy corn? You're not going to eat it all, right? So you don't want to keep making it or buying it. So the product, candy corn, is actually going to feed back, and when the allosteric site is open, so see how it's closed right now because there's a substrate bound? If there wasn't a substrate bound, it'd be open, allow this product to bind, changes the shape of the enzyme, closes the active site, you can't make candy corn anymore. Eventually, once you start to run out of it, and if you want to make more, this would be removed, the enzyme would go back to its normal shape, and you could start making it again. So this is being done on purpose, generally in a multi-step pathway, not just one enzyme. Usually you're looking at some type of chemical pathway that's using like 2, 3, 4 enzymes to make a product. That product will feed back and shut down its own production. Now, why are you doing this? To maintain homeostasis, to conserve energy. Why are you going to keep making something like insulin? We use that example so many times from the beginning of the year, if you already have enough of it. So that product can feed back and shut down its own production and restart itself when its numbers start to run low. All so you can maintain homeostasis, not waste energy, and not throw your body out of whack. All right, so again, feedback inhibition, going to be a form of allosteric inhibition, but it's when you are using the product as the actual inhibitor. And here's a graphic to show you that. So if you just focus down here, again, we have 3 enzymes, 1, 2, 3, who are trying to turn a green triangle eventually into the product of a yellow square. All right, so you can see how you have intermediates until you get to the final product. So the product of enzyme 1 is the substrate of enzyme 2 and vice versa. If you make too much of this final product here, it could feed back, usually to the first enzyme in the pathway, act as the allosteric inhibitor, change the shape of the active site. It's not a triangle anymore, it's like a little square. Substrate can't bind, you've shut down the pathway. So again, this is a good way to maintain homeostasis within a cell, an organ, or the whole body altogether. To finish up, we can watch this in action.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways,
so we have 4 enzymes that are trying to turn a substrate eventually into the final product over here.
The product of one reaction becomes the substrate for the next reaction.
There's your final product.
If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site.
So here comes the product that we have too much of. It's going to come to the first enzyme, bind to the allosteric site. Watch what happens to the active site.
When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So now you have too much of this product. It's actually shut itself down. If you start to run out of this product, eventually there would probably be another enzyme whose job is to come and remove this from the allosteric site. And again, once that's free, the whole process would start again.
Cell, such as those involved.
All right, without listening to it, we can just drag it through, and the whole process would pick up again. So it's a back and forth. So right now you have a positive feedback loop where you're turning on a system and making a final product, but eventually in this instance for feedback, the product can now move away, bind to the allosteric site of the first enzyme, and now create a negative feedback loop where you shut down a system, which is going to be the formation of this product. So not all inhibitors are bad, all right? Some are good, some are bad. All right, in this instance, this is an example of how your body has evolved to shut down its own system and maintain homeostasis. If you have any questions, make sure to ask during our live class. Thank you.